So this assignment and quiz will be due a week. And we'll give you a week to do this on the 7th. Because every time I screw up, it might take you 6 hours to find out. So that does not mean wait for 4 or 5 days to start the assignment. That means if you start it now, it probably is enough time to finish. If you manage to get it right the first time, that's definitely enough to finish. But what are the odds of that, right? So we'll go through what it looks like, and we're basically going to do the framework of it today in class. So it shouldn't be anything like new or scary. All right. So yeah, let's start out here with public genomic data, because there's a crap ton of public genomic data out there, super useful, and you're going to find yourself needing it all the time. So you're working in cancer research, and you have some new cells, and you sequence these cells. What do you do with that data? Get your tumor cells here. They're showing that this drug is useful on them. Okay, why? What do you do? Great, you just sequenced it. Problem solved? No, problem just starting at that point. What's different about them that makes this drug effective? Yeah, sounds like you got an idea.
You might see the variant. See what kind of like genes or interests are different in the tumor cells versus like a standard, or maybe like a standard for like the reference genome of interest.
So yeah, this is super common. It's like we've got something interesting. It's a variant. And we go, okay, well, how is it different? What genes are different from like basic human genome? That's a really common kind of workflow, is we're comparing one genome to another to look for differences. And there's whole branches of bioinformatics, genome-wide association studies. You could have 1,000 genomes from a population of people, some with a disease and some without. And you're asking the question, is it their genes that's causing the disease? Is it environment? Is it a combination of the two? And so you look at what's different between all their genomes. Are there any genes being expressed statistically significantly different between the two populations? Okay, well, you have to compare against the human reference genome. So boom, you're talking public data right there. Now, there's also this other problem that every time you sequence the genome of some cells or something, the odds that you were perfectly sterile are effectively 0. And this is what assignment 7 is. It's finding dog DNA in things where it shouldn't be. I find dog DNA in coral reef samples. Why? Because the grad students who were extracting DNA have dogs at home. It's that simple. Recently went to a talk where someone was describing the variety of Saccharomyces cerevisiae, the bread yeast, brewer's yeast, found in the rocks. So he was like working with oil companies, and they're drilling, and they like refit with sterile drills and bring up like deep earth rock samples. And he's like, yeah, there's yeast down there. No, there's not. You brew for a hobby. You were just talking about that like an hour ago. That's what you're finding. So, okay, we're looking for contaminants. So we need the yeast genome, we need dog genome, all that stuff. Again, public DNA, right? And so there's the NCBI, which surely you've heard of. I hope so. Okay. What's your familiarity with like the NCBI website, running blast? Stuff? Accession numbers. Well, we're going to be playing around on it. The sequence read archive, SRA here, is where all the short reads go. So this is not where somebody would put like their freshly assembled dog genome that they're excited about. This is for like just that raw data. Raw data we release. So we're gonna take a look at that. And then we're gonna use a tool called SRA Toolkit, which is provided by NCBI to access that data programmatically. And then we're gonna also use this other package called datasets. And that's provided by NCBI. And that's more for accessing like those whole genomes and not the raw data, but like assembled good quality stuff. And then we're going to do kind of exactly what you're talking about. We're going to compare sequences to references. This is what BBMap was originally created for. So we've used that before. But BBMap is for taking a reference genome. I mean, it does more than this now. It takes short reads and it maps them and says where they belong. So we're going to do that. And then it's going to create what's called a SAM file, which is a very annoying file type, but one that you're going to have to deal with a lot. That's sequence alignment map, SAM. So that's the output of I mapped my short sequences onto this reference genome. So it's just a tab limited thing, but it's kind of hard to read. In fact, it's hard enough to read that an entire separate software package was created to deal with SAM files called SAM tools. So we'll look at using SAM tools to kind of like explore what's going on. Okay, so let's go to SRA. Sequence read archive. Also, because of the lapse of government funding, we don't have a functioning society right now. We've blocked a bunch of ads for you. And this may be inaccurate, it's not being updated, but we're okay. Assuming civilization doesn't collapse, this will be back and updated again. Currently, we can still search the old version. All right, so we want to find, for our purposes today, somebody's short Illumina reads from somewhere. And we're going to also download an E. coli reference genome and kind of look for E. coli sequences in their short reads. So we do.
You just pick a random place.
We can pick a random place. We can pick random sample type. We can just start searching. I don't care what it is right now. We'll refine it once we get to the results page. But
It's like a weird one.
I'm open to suggestion.
I know when I was like, I don't know, I continued to like tooth medicines, like black, it was black.
Okay. So we've got dental plaque. We've got results by taxon. This can be kind of helpful over here. Then we've got a whole list of filters over here on the left. And then super handy is down here. This shows you like the actual what you searched. So if you are going to interact with this using the command line, which you can do, there's another package for that. So you don't have to go online and do this. And you don't want to have to formulate this will like kind of print out all your searching. All right, so we've got dental plaque, human oral metagenome, or just oral metagenome. We got Homo sapiens, human metagenome. You can look at one of these. Illumina Nova C 6000 paired end sequencing. That is not a very good sample title. It's terrible, but that's what people do. Dental plaque microbiomes from hunter-gatherer and subsistence farmer populations of Cameroon. That's pretty cool, actually. But the reason I don't like this one is because it's huge. 20 gigabytes for this one sample. But that is really cool. I want to do it.
It's they're all gonna do it.
Okay. Well, this tells us a lot about it. We've got source metagenomic. What's that mean?
They're just experiencing everything in there.
Everything, right? So they didn't like culture just the human cells or culture some bacteria or whatever. They're just like, here's a, we scrape some people's teeth and we sequence everything. It's the metagenome. Layout, paired. So we got forward and reverse. That tracks with the Illumina NovaSeq there. Selection, random. You might also see amplicon there. And that gets at this little right here, amplicon versus shotgun or random sequencing. These have both been called metagenomic. Amplicon is where you amplify a single gene. So it might be 16S if you're interested in just bacteria. Might be 18S if you're just interested in eukaryotes. If it's fungi, maybe you're going for the ITS, that internal transcribe spacer. But like what we're talking about is we're looking for just one type of organism. We've got primers that will just amplify that one. So we just amplify that, and that's what we're seeing. Versus shotgun metagenome. This is where you don't do any of that. Use random DNA fragments from the entire sample. So there's no selection happening. And there's a number of different ways people do this. They use random primers. So these little hexamers, 6 nucleotides, every possible combination of 6 nucleotides gets thrown into a tube as primers. So it amplifies everything. Or there's like single genome amplification where I don't understand the chemistry, but it just amplifies like from a single cell. The entire genome kind of randomly. Pros and cons for amplicon versus shotgun.
There's probably more.
So for fungi that I work with a lot, there's good databases of ITS sequences linked to names and taxonomy and all that stuff. Because we don't have whole genomes from all these fungi. People have just been sequencing this ITS for decades now. So that's kind of built up. So that's really nice. If I'm interested in fungi and I do shotgun sequencing of like a leaf sample, I just want to know what fungi are in there. 99% of the DNA that I sequence will be that leaf. So 1% of my data will be useful. So it's pretty inefficient. So it costs a lot more if you're just looking for a certain thing. I'd have to do like 100 times deeper sequencing to find the fungi in the leaf sample. But shotgun has benefits. It's the only way to find unknown organisms. Amplicon depends on having primers that work for a certain group. And we've seen this in fungi that our universal primers for fungi don't actually amplify everything. So if we want to see what fungi are there in the ocean, we can't use amplicon sequencing. We have to do shotgun. We just have to deal with the fact that at least half of it's going to be bacterial that we throw away for our study. Then again, there's what if I actually want to know everything? I want to know all the bacteria and the fungi and the archaea and the protists and the plant. I want to see everything. Shotgun's the way to go for that. So this one, random, that's referring to shotgun. There's no selection happening here. There's no like amplification. So we're gonna get all kinds of crap. Is there one that's maybe more than 20 gigs? That's so huge.
Can you filter based on size or?
Let's see.
It's 1.7.
Megabytes?
Wait a second.
Yeah, that's a little too small. Yeah. Yeah, we can start filtering here. So one thing, we don't want BAM files, we don't want SFF. So we can just say, all right, let's limit to FASTQ. We can go over here and say, yeah, just the human oral metagenome. Look what it's doing to our search. It's populating it with all the filters. So we can be reproducible. So keep that in mind. If you're doing this filtering and you're like finding samples to use for your study, the way to make it reproducible is to copy and paste that into your README or wherever. We can make it part of our command line workflow eventually, but. All right. We definitely want Illumina. We want paired in for reverse. We don't want RNA, we want DNA for our question here. Show additional filters. Other. Oh, that's not helpful. Okay, but we're down to 7,831 results, which is pretty small for SRA. We started with a couple 100,000. So up here at the top, we can send results to run select. All right, now this is where we can really start filtering. So this gives us all 7,831 kind of filtered down to, and it actually gives us metadata about them. So we can see, look, the number of bases, the size of the file. You see where it was sequenced, the instrument it was sequenced on, all that library selection. Oh, we got all kinds of stuff in here. Host family relationship. So somewhere in our data, somebody has recorded this information, which is kind of cool. Might not be hunter-gatherer stuff, which is less exciting, but we can filter. So we can go up here and filter on bytes. Okay, so here we go. How big is the file that we want? 0 to 5 gigs. That's more like it for running on the login node. If we wanted to just find those drinks, oh, host eat candy or not? Oh, fun. Yes, candy. Okay, suddenly we're like looking at a very different sort of situation here. We got Latin longitude, host age, dental plaque. Gender, whether they floss or not. So this is somebody's like actual study. I'm just going to go into it to show you. Bioproject is where the study lives, and you can see all those numbers are the same. So these are all coming from like the same paper, essentially. Okay. Characterize oral microbiome and HIV infection and periodontal disease. Sequencing data includes 16S B34 amplicon. So this is not shotgun. They have amplified just the bacterial B3 through B4 region of the 16S. So that's pretty cool, and that's why the file is a lot smaller, because it's just that amplicon data. But we don't want this for our purposes.
Did we navigate like through the thing and it said earlier, it said random, and now it says.
No, we didn't say random.
Like on the.
We said metagenome, and both of these are considered metagenome, 16 assets.
And then like on the, like when you're on the actual page for like, like the first one, before we got to the unselected,
We go back.
like on the, we just click on the, that top one.
Okay, so here's what we've done. Human oral metagenome, it needs to be DNA, paired library, Illumina, file type, FASTQ, that's it.
Okay. And so this is like, when you go to run selector, that's all 7,000.
Mhm. Or whichever ones that you like highlight here. Yeah. So my workflow is I kind of get it down using search here. I record what I've searched for reproducibility, send to run selector, and that's when you can really start honing in. At this point, I honestly just export this whole thing, use like R to filter, and select what I want. But we want bytes, we want smaller stuff. And here's where we'll be able to filter for like shotgun versus amp. There, library selection. How did we select this library? So we've got PCR, we've got random. We want random. Unspecified means these assholes didn't even write it down. That makes their data useless. So we've got random. Cool. This ain't so bad. These are smaller files. Let's see what this is all about. 90 megabytes. Yeah, okay, so the same study. Cameron, Baca, and Zime. 2 plaque samples collected per individual, anterior and posterior teeth. Okay, this will be perfect. So this is the sample I want, but maybe I want the whole study. I want everything from that. What I'm gonna do is right here, just click the metadata button. And that downloads the file for me. sRA run table.csv. Right? Neat. There it is. So I've got a nice like spreadsheet version of everything. I've got their latitude and longitude, subject ID. For our purposes, we're only going to be doing that first one. But this is the column that we're interested in, the run. That's where our accessions are.
So should we just write down cause I don't know, so I don't have this
Oh.
file, but
Yeah,
should I like?
good thing we're recording, but yeah, so you get this file by clicking metadata here.
Wait, like getting. Well, I can like redo this, but are we just doing the one accession number today?
Just today,
So should I
yeah,
just for this purpose write down that accession number?
yeah, let's do that. So
Sorry.
that's okay, no worries. So, you know, in a real situation, you probably want more than one, and so you're gonna be exporting this metadata table. So I'm doing this recently for one of my projects. I told you about the marine fungi thing. So I've went and found every study out there that was shotgun metagenomes of ocean water of some sort, essentially. I downloaded that whole table, and then I've got this list of accession numbers. So then I can use the tools we're about to install, download each of those by their accession, run that pipeline up. So this is the one I think we wanted, because it's kind of small. It's that hunter-gatherer dental plaque, and so. I clicked metadata that downloaded it as a CSV. That CSV gets put right on our HPC into our project. So our lesson 7 folder, it goes in there as the metadata, and this is what it looks like. But it didn't put it in the order that we were showing it there. It put it in its own. So that's the one we want. I want to make sure I can find it here in the file. What?
Is it for the biosimple or is it?
It's the run.
And by like getting the wrong, you'll get that accession.
That's the accession. So in the sequence read archive, the run column is the accession. A unique identifier for that sample.
Okay, down to the wrong.
No, you're looking good. I just clicked that. So that way we're getting just the smaller files. Yeah. Okay, so that's the same, just click on run the other way. There. See right here, so you can filter and you copy this. Or you can rearrange. That's just alphabetical. I could rearrange here by the number of bytes. Right, smallest to largest, largest to smallest. So here we go. ERR. This will be our thing for today.
Is there a reason?
Well, that one's so tiny. 90 megs. That's basically a failed sample. Let's go with the second one here. Experience is telling me that we're not going to find OI in that person. It's failed. So ERR 1008 3819. Yeah, there it is in that file I downloaded. Yeah. So for assignment 7, I'm gonna have you pick at least 10 samples somehow from SRA and download this Excel spreadsheet and then do what we're about to do in a for loop. You're looping through that first column. It's not a big conceptual loop. Yeah.
So are we gonna have to like get the CSV onto it somehow?
Somehow. What do you mean somehow? You know how.
Personally.
FileZilla. I like FileZilla too. Yeah, it's good. It's got some like adware in it, but I'm cool with it because up and drag is beautiful. Yeah, so FTP, FileZilla, however. Yeah. And so yeah, this will you take this as like the starting point. This is where I start. So if I want somebody to reproduce my entire assignment 7, you can give them the directory structure, my pipeline, and this file.
And then like for the purposes like are we just gonna have like all of this in here, or are we just gonna have like the one column of like the run number, like
We
the?
want everything because we might want that, right? There's going to be a column in there that says whether it was the front or back of their mouth. Maybe that's part of your question, or their age or whatever. So you want that available. But for assignment 7, no.
Okay,
So we're just going to be picking that first column and looking through those accessions. So these are our accessions here. Each one will be unique. But for this lesson, we're just picking that one, ERR 1008 3819. So I've got that copied, ready to go. Now I'm gonna connect to Aura. Yeah.
Sorry, all of those copied.
No, just that one.
Okay.
So for our lesson, we're all working with the same sample. So that same accession number. And Laura already. No. Go into lesson 7. Got our first job in there. It's all kind of messy, so I'm gonna clean all that up, remove everything in there. Fresh start. Make some directories. Data. Ref. Output. Oh, and scripts. In data, I'm going to nano accession.txt file and paste in that. So I've got a nice little reference. Don't have that memorized.
So for this one, could we hypothetically just do that with our accession numbers, or do you want us to use this?
Use the CSV. Maybe I'll just do it that way. Just to show you. And copy that whole line. Paste it in. The only thing we need in order to download this is that accession over there.
So then when we're doing ours, like we'll look at the CSV and it's gonna be just like, all right, let's just say like for 20 samples, each like column is like comma separated, so then like when we're doing the report, we just want like the first.
Yeah, just the first column. And you know how to do that with cut and some process substitution. So it'll look like that, whatever.
Should put, okay, cause it'll have the like the number.
It will. What we do there. Tail n +1 or +2 starts on the second row. Cut 1, boom. So we've got that to loop through. You haven't changed your metadata whatsoever. You just use the process substitution. All that old stuff in the past 2 months. Okay, so we've got that. I'll save it. So our accession is stored there. Inside of data. We're going to have output, refs, and scripts. And I need to go ahead and document before I forget. Oops. What my search term was for this. There we go. According to the date, because the Sequence Read Archive is updated all the time when we have a government. It's not in. That's a big, ugly, long search term that I would not want to have to have typed out. My own, but there you have it.
What is that like search term? That doesn't include filtered out of like the.
Right.
So you have to like document that separately or.
But honestly, all you need is that SRA run tables. Hey, here's the stuff I did. This is just kind of like a reminder for other people, like, well, how'd you get this? Well, here's what I searched. Okay. All right. So now we've got a README data with our accessions in it. All right, now all we need is to actually get the tools for downloading this stuff. So here we are. Get this installed. The SRA toolkit. Are none of these. We have Fedora Red Hat. That's what the HPC is running.
It sounds like
Fedora, right?
Mhm.
Someone's named it that.
Yes, they did. So what do? Ubuntu tends to be the most compatible with everything. I'm just gonna go right into my programs. Like I've already installed it here. Remove our SRA toolkit. Okay,
How'd you use Kotlin versus Rose?
I'm just trying shit.
Okay. Like,
Um,
if you have an action.
But you're not running a Mac, you're on HPC.
Huh.
So you're on Fedora, Red Hat, Linux. Sorry to say. Fedora and Ubuntu come from the same lineage in Linux world, and so that's where I'm gonna go. For X C F. Sorry, toolkit. So I downloaded it.
What's the tar IC?
Tar is for tarball. That's this part of the file here. So it's kind of like a zip. It's all smashed together, a bunch of files. And so the X is for extract. Z is for, hey, this used gzip to compress everything. I know that because of the .gz, or I assume that because of the .gz. And then F is the file. Imagine like somebody with a thick German accent, Werner Herzog, saying, extract the file. That's typically how I think of it in my head because I'm a freak. But XZF, extract the file. Well, what that did was give me that folder. And here's the leftover tarball. So I'm going to remove that, clean up. I'm going to go into SRA toolkit. And look, it's got a bin. I'm betting everything's in the bin. Yeah, whole bunch of stuff sitting there in the bin. The one we care about is faster queue dump. There's a whole bunch of stuff in here. It can come in handy, but I think 99% of the time, this is what everybody cares about for this SRA toolkit. It's that. So. This PWD. Is where. Faster Q dump lives, so I need to do what with that?
Toward path.
Yeah, what is my path? So nano home dot bash RC. Scroll down. There, got it. That's where path equals that. And then a nice exec bash. Reset and get it loaded, and from anywhere, from my home, should be able to say which faster Q dash dump. Yay, found it. Cool. Hopefully you guys are getting really used to this install add the path stuff at this point. So we've got the SRA toolkit. And it works really easily. Faster Q dump, and then you just give it the accession number. That's it. And wherever you are, you'll download those sequence files associated with that accession. So where do I want to be?
Lesson 5.
Lesson 7 data, yeah. I'm gonna go back into biocomputing, lesson 7 data. Faster Q dump. And I need to feed it that accession somehow. From my file. How can I get that? My file's sitting here in accession.txt. Cut field 1. Boom. There it is. So if I echo that, yep. So I should just be able to say faster Q dump cat accession.txt text cut. Make sense? Using those tricksy Linux commands. And it's doing its thing. This would be real easy in a for loop. Where I in that do faster Q dump I done. It will sit there and download every single accession that you've got in your like metadata file. This one was like what, 100 megabytes or something, not too big. Close to a gig. Find it here. There it is in bytes. I can't do 1024. Not in my head. But it's downloading. Yes.
I think it's regular.
Yeah, it's not. You did exact that. So let's do at the dash RC. That's completely.
Oh, that's pretty like the.
Oh, yeah. You need to clean that up, but okay. Yeah, it's not everything. So, you know. Dash RC.
I put it above that.
Oh, okay, you did.
Oh.
Oh, yeah.
Was it big
Yeah.
long to run?
Hmm?
Was it big long to run? I just did it faster, you know.
I mean, mine's finished. When you see this output. And it's
I
done.
got very. Way less. I could have done
With this,
it. No, it's not. Everyone.
that would be
It's like a little.
Yeah, I got this. Is it the same one that we had earlier?
these are all the same. Uh, is it actually there?
I'm lazy, I mean, I didn't do it.
Mhm. Yeah, it's working, it's there. Export command not found. Not when you're in a conda environment. Conda deactivate. Boom, boom. Bites again. Hate conda. Okay. Conda's awesome. I'm being whiny, but it does crack like that.
Like it's.
Export. So every time you log in, it's putting in Conda.
I think they're really good.
Right. Two export. So it's not like even the one that copy paste. So export does work.
Is it supposed to be a
Oh,
Yeah.
yay. Okay. Programs, that's how they have a tool for that. Okay. Got it.
Thank you so much, sorry about that.
Sorry. All right, is everybody's downloaded? Okay, neat, that's SRA toolkit. Congratulations. You can get anything from the sequence read archive if you know its succession. Works great with while loops and for loops and everything, and it just downloads it right to where you are. There's a whole lot more you can do with SRA toolkit, which we'll skip over for now. All right, time for another download. We want data sets. Good. So that's this link here. Data sets. So there's the download button, but I don't want to click it because that'll download it to my computer, because here I am. So I right click. Copy the link. Or wait, look how nice they are. Right there. Command line instructions for us. So I'm going back into programs. I'm going to paste that in. That's quick. Since programs is already in my path, I'm not going to have to do anything else except make this executable. Chmod plus X data sets. All done. Okay, well, that was exciting. So data sets works a bit differently. It's data set. So look, I'm just going to go back to my biocomputing. I'm done installing software now. Lesson 7. Yes. Yeah, Chmod plus X. So anywhere in my terminal now, I can just say data sets. And it gives me a few options: summary, download, or dehydrate. Or rehydrate. Data sets, download. This works just like Git, just like CTK, where it's like name of the program, name of the subprogram. Okay, so data sets download says, all right, cool. Do you want a gene, a genome, a taxonomy, or a virus? Data sets download genome. Great. So it says, do you want to go by accession number or by taxon? Well, taxon. All right, well, it gives you some examples here. Data sets download genome taxon human. Very simple. You can tell it which chromosomes to include. You can give it a scientific name in quotes. You can say, just give me the reference genome. How do I use this? Well, it tells you right there. Data sets download genome taxon command help. Okay. Genome taxon -- help. Okay, limit it to annotated genomes. We can have an API key if we're like doing thousands of these. NCBI will absolutely think we're a bot. So we have to have like a pre-registered key saying I'm a real person. We can say what we want, just the genome, the nucleotide coding sequences only. We want this in general feature format. So there's a lot we can do. We can say limit it to reference genomes. We can give it dates, at least before and after. All right, but what do we want for our lesson? E. coli. So right there, I'm going to give it the full scientific name, and I'm going to say reference and then the file name. So I'm just going to copy paste this. And then the second line or third line, it looks like here, that wraps unzip D is a ref.
What's the
Directory for unzip, like where to unzip something. So we're downloading the E. coli reference genome. We're naming it ref/E. coli.zip, and then we're going to unzip it into that reference. So I copy this. I want to run it straight from my lesson 5 or lesson 7. Done. Now if I look around the tree, I've got my accession number stored in this file. I've got the 2 accessions I downloaded, or the 1 accession for reverse reads. And then in my reference, I've got a subfolder called NCBI dataset, which has a subfolder called data, which has 2 versions of the E. coli genome. Let's just go with this first one. But it's kind of hard to get there. You have to go into ref, the NCBI dataset, data, GCF, blah, 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 and there's the file. Okay. We've all got BBMap already in our path, and so now we're ready to run it. We're going to take our accessions forward and reverse, map them against E. coli. And what is the question that we're actually asking here?
is the one from the, eventually. Yeah.
Is E. coli found in the dental plaque? Yep, of this particular sample. This won't quite answer it yet for us, but it'll get us the files that will answer it for us. This is a believable sort of thing. E. coli turns up all the time where we're not actually interested in it because, you know, it's like usually a contaminant because people don't wash their hands well. And so it's just kind of around in the lab. Anywhere humans are, E. coli is bound to follow. And so very often, if I'm doing coral reef work, I map it against E. coli and I find E. coli and I remove those sequences because they're contaminants very clearly. If I see dog in there, I remove them. If I see human genome stuff, I remove it. So we're looking for contaminants, at least in this little step. So BB, yeah.
How would you, I guess, how do you decide how many contaminants are found?
All those, the common ones is like cat, dog, human, E. coli, and then blanking on the name of the yeast. It's on our skin. No. I should know this. It's literally my job. I'm just like, but so there's like a, you know, top 10 most wanted other things. They turn up all the time, everywhere, and that's not real. All right, so BB map. We've seen it before, but we have a reference. Oh, we haven't used BB map. We use BB stats, right? Okay. So just like in BB stats, it's the the way that it's written is the argument equals no space between that and then that value for the argument. So the reference, we're gonna have to make that our E. coli reference. In1 equals, that'll be our forward read. In2 equals will be our reverse read, and then output. Okay, so we need to point this to our actual reference where it is. And the actual forward and reverse reads where they are, they're just in data. So that's nice and easy. But our reference genome is kind of buried in a bunch of subfolders. So if I try to find it, I can use LL just to give me that tab complete magic. I can go into ref, NCBI dataset, data, then, oh, what do I want? These, yeah. We'll do that one that starts with 5. So that whole long ass file path is the relative path to our E. coli reference genome. So then I hit home, delete LL, and just say capital ref equals, and I'll put that in quotes. Making myself a nice little variable. Definitely want to document that into my README. Because I'm kind of building my pipeline here. So now I can just call on ref anytime I want with $sign. Let's look at the E. coli genome. Woo! There it is. Hmm.
Can you back up?
Or do you want the tree or?
Um, Can you show me like what you did, like the side and then like to call it?
Right, it's some PWD for that, right, you have to do. You have to get like the thought matters, right, just.
I don't understand the question.
Do you have to like add it?
Like, okay, so you signed rough, and then how did you do the next thing to like see the data in rough?
Oh,
Yeah, I don't know if I should do it, but
Cat ref.
Okay, oh, cat.
so this is me just testing to make sure that file actually exists. I assigned that variable correctly. If I can cat it and it doesn't say file not found, then I'm happy.
It doesn't matter which one they are.
I went with the first one. There, yeah. We have 2 complete reference genomes for E. coli. There's only a few base pair differences, I think. So cat ref. Word count C does what? So I'm just getting an estimate on the size of this genome. It's about right for E. coli. I'm happy. In the ballpark of what I think of. I think of E. coli genome size. Okay. Well, we go to our BB map. And we need to tell it our reference genome, which is $ref. And then we need to tell it N1, which is data. That first fast view. We need to tell it our N2, our second read, which is data. Second FASTQ. Then we need to tell it what out. And what did I call it in the lesson? Oh yeah, output mapped to coli.sam. And then I'm also going to give it a min ID of 0.95. There's a million things you can do to determine how BBMap matches sequences up against a reference. We're keeping this real simple for us and saying it's got to have 95% identity. And that's it. Boom.
I got, no.
Say,
Yeah,
I don't think I have.
I think, did we sound things like what, did we just do the content? Yeah, I don't know.
Did you just do it in Conda?
I think
I
so.
think we did.
Oh, well, go ahead and activate that Conda environment and then you're good to go. Conda activate BBMap and
That's what is the high level.
I think that would be.
whoa.
Like my session, I only have the session.
Okay. That's fine.
No,
Yeah, your stuff never finished downloading.
yeah, so, wait, what's the?
So those are still the temporary download
Oh,
spots.
so much.
So you need to go,
So should we like take?
yeah. So you don't have the accession numbers in your data. Notice how I've got these two files. You don't have them because they didn't finish downloading. They're still in the .c temp. So you'd have to start over, download, run that download again. Okay. And it'll take a few minutes.
Okay.
All right, maybe it's easier if you just hurry and install BBMap locally style instead of Conda. Judging from everybody's like README files in your commentary about the last assignment, nobody seemed to really love Conda. I'm with you there. I prefer a local install because I have control. It's a bit scary, but you know how to do it. You just did it twice. So,
I have a question, because we're trying to do the modular way. And I keep saying run Conda and before Conda. I was saying earlier.
don't do that.
No, yeah, I did it.
Do you remember that old line, that whole like
Yeah.
source the,
Yeah.
Yeah. then you can Conda activate,
It's like the module mini forge source and then you do contact activate.
and BBMap end.
Yeah.
Oh, for BBMap. Yeah, it's working. Don't stop it.
Okay.
Yeah.
Yeah.
It's like a few gigs, so like that.
Okay, I just thought something got wrong with the.
Y'all are noticing like the pace of this class starting to pick up, that's on purpose. It's not because I hate you. Because you've done nearly everything here before, except for on the because I believe in you. All right, we got all kinds of nonsense in here now. After mine ran, it went in, it's got all this like scaffolds and summary and all that stuff, but the output file that we told it to make, that's there, so I'm happy. If I really care, I can start deleting that stuff when I am finished with this pipeline to clean it up and not take up so much room. But that's all part of BB map. And forward that for posterity. I'm gonna try to find my command I used to download the accessions. So I'm just gonna say history. Faster Q. Oh, grep, faster Q, duh. There it is. I'm searching through my history, finding faster Q. Now go here. So that's download my accessions from SRA. I'm gonna grep through my history and find data sets, and there it is, my command that I used for that. Up here is the handy unzip command. Okay. Yes.
I got an error when I was trying to run that command, that says, there's a no reference. Does that mean that like my app, like my variable of my reference?
One of your, it's probably your ref. And so try echo $ref.
Okay,
Is it empty or?
yeah, it's empty.
Yeah, so you have to make sure that that variable is set. Did you have a space when you set the equals or anything?
Okay, I just downloaded I think I just didn't call the variable.
Oh, okay.
I reactivated the thing, not really good.
Yeah.
Yeah, it works.
Okay. Oh, yeah. There we go. All right, so far, really all we've done is download those accessions. We've got this metadata sheet that would have to come with this project. If I gave this to somebody, that's part of my reproducible methods. Like, here's the accession sheet I'm starting with. Okay, and Since we're only doing one, we don't have to do a for loop there. We're downloading that. Then we're using data sets to get E. coli. Unzipping it, putting it where it belongs. Setting a variable to point to that E. coli reference genome. And then bbmap. We're taking our 2 downloaded accessions. 1 accession, 2 downloaded files for that accession. Mapping them against the reference E. coli genome. And the output is going into output. There it is, and it's a huge, stupid file. It exploded to 5.1 gigs. Whoops. You know what? And just give me the first 2 lines. No. First 3. No, those are all header lines. There we go. Okay. So that's 1 line of a SAM file. Sequence alignment map. And it's saying, here's the name of the sequence that I tried to map. Okay. So it's basically just the header of that sequence. And then a whole bunch of information about it, including that sequence itself and its quality scores. Well, there's no need to memorize what that is. Let me show you. Oh, like that's readable. Yeah, okay. So we got the name, some flag. This one's 99. The name of the reference that we mapped it to, and then the coordinates of where it mapped. So starting at position 7, ending at 30. Okay. That's not so crazy. If you look at this right here, it's showing you that there was this short read that mapped onto the reference genome, starting at position 7, ending at position 30. And so that's really what's going on here, is that they've tried to find some like tab delimited way of showing that map of like where all the sequences we had map onto that original genome. And so they've come up with, these brilliant psychopaths came up with this impossible to read format that also includes this idea of a flag, includes alignment information about the read. Okay. The flags are binary. So if there's 0, that means we got zeros all the way across here, and that just means the read got mapped, it's happy, the forward strand, here it is. If there's a 1 in this position, that's binary for 1, means it's part of a paired end read. If it's got a 1 in this position, which is binary for 2, properly paired. 1 in this position was binary for 4, read is unmapped, so it didn't find a hit. This one, the mate is unmapped, so the opposite read. So there's like all this information that we can learn about it, and it's all condensed into this little binary format. And here's the thing, is that they're additive. So if you have 0, 0, 0, 0, 0, 1, 1, 0, 0, 0, that means it's mapped to the reverse strand and the mate is unmapped. What this means is that we just put numbers together in 16 right here. It's mapped to the reverse strand, so it's reverse complemented. Flags 4 and 16, which 20, so you can see a 20 in that position, and that means it's mapped to the reverse strand and unmapped. The mate is mapped to the reverse strand. Confused yet? Yeah. So there's a lot that can go on when you're taking these reads against a reference genome and you're flipping them both ways and doing reverse complement and trying to find if they match up against there anyway, like anywhere in any position, and then whether the paired reverse read did the same thing or if it was in like reverse complement. A perfect match, imagine a genome stretching out long ways. You've got a forward and reverse read that it should be reverse complements of each other. They should both be mapped up against their one in the opposite orientation. So that's a perfect match, 100% alignment, all that good stuff. That just gets a 0 flag. Well, what do we see here is this particular read has a flag of 77. I don't know what that means. I don't want to have to go look it up and do the binary maths to figure it out either. So I use SAM tools. We don't have time to get that installed today, but SAM tools lets us not care about any of that stuff that I talked about and use like some basic little like, let's filter this. So I can view that. I can find the ones that have a flag of 4, meaning they did map up. Look at that. All of these reads map to E. coli. There's poop on those teeth. A lot of it. Right, the lowercase f gives you the opposite. Yeah. All right. Sanity check. Yeah, capital F is the opposite of unmapped. There's still a bunch of poop on those teeth, not as much as I thought. Whew. Okay, well,
Did you?
it's in the lesson also. So we almost got through it today. That's all right. We've got next time as well, but go ahead and start assignment 7. It's going to have you doing what we just did, but with 10 or more, exactly. Whatever you find. Now, do not run that on the login node. You will get put in timeout, like literally they will restrict your account and say you can't use this for an hour or a day or whatever they decide. It gets submitted as a SLARM job, so that whole the downloading and the downloading of the reference genome, you can do that outside of the SLARM job. I'm not worried about that. But once you get to the DB map part, that part needs to be a SLARM job. And you can do that via Conda or local install. A week from today.